ever so much for the introduction and also the invitation to come and speak to you this evening. I should say in response to John that we've also solved the problem about initials. We just, when we're shortlisting candidates for new academic positions, we just make sure that none of them have got my initials, and then that solves that problem. Actually, that's probably not legal, is it? Anyway, enough of this. I wanted to introduce you to a story that you probably know a lot about already, but I don't want to rake over the last year in too much detail. I want to make sure you can see the correct screen. You can see the whole screen, can't you, with my presentation? Yeah, good. Usually there's a green thing around the outside and it's disappeared. Okay, so what I wanted to do was, there we go. Sorry, I'm just reorganizing things. To talk to you about a problem that emerged last year, and it continues, and it's associated with a problem you'll be very familiar with, the COVID crisis that we've been living through over the last year. What I didn't want to do is to sort of talk to you too much about what you probably heard a lot about already. I just wanted to focus on an interesting phenomenon and also very worrying phenomenon concerning blood clotting, but we'll come to that in a moment. So the last year or 18 months has been a really interesting period because it's a period where the general public has become very familiar with biology and medical science. They know what viruses are, they know how they work, at least in some level. They know some of the problems that occur when you get infections and the seriousness of this. And so we're sort of living through interesting times where the sort of public interest and understanding of what we do has been transformed. And I think that's one of the really interesting things I've noticed in the last year, particularly with the area I work in, which I'll come to you in a moment. We've all got familiar with looking at these rather worrying diagrams on the 10 o'clock news or one of the government press conferences. In this case, looking at the death rate. So this shows you that, so this goes up to the end of February. So this shows you the death rates over that period up until the end of February, and really the quite worrying picture that emerged. And really unprecedented times in terms of this type of health crisis and the speed in which we've had to adapt and adjust. But the other thing that's been really interesting for me over the last year is the way that scientists and doctors and clinician scientists have started to work together in different ways. Probably in many cases, interactions that may have happened anyway in time, but we've been forced into this situation where we need to solve some really urgent problems very quickly. And I guess one of the things that really struck me quite early on in the COVID crisis, when we went into the first lockdown last year, was how much time I spent talking to talking to clinicians who really didn't know what to do. There was a genuine sense of panic, particularly as the symptoms of this particular disease are so complex, multifactorial, and all sorts of different ideas and theories out there. So it's been a period of really, I felt like I've actually had something to contribute to sort of frontline medical science, if you like, rather than it being somewhere in the dim and distant future, you know, associated with translation of some of the research we do normally. It's enabled us to really contribute to the development of stories as they evolve very, very quickly in front of us in a very public manner. And what I'm going to do is to tell you one or two stories that fit around this and things that we've been doing over the last year. But this all starts with this little beast. This is a coronavirus. This is the SARS-CoV-2 virus, a cartoon of it at least. And really just the features that I'm going to come back to once or twice as we go through is particularly this purple protein on the outside, the spike protein as it's often referred to. But you can see the inside, that there are other proteins actually within the outside wall of this virus. I should point out, before we go too far actually, that everything I know about microbiology, John Granger taught me. So if there's anything wrong with any of what I'm showing you at the moment, he's the man to blame. So this is a single-stranded RNA virus, and so that's the message, if you like, inside the virus that enables the genes to be translated in the infected cells, which we'll come to in a little while. And this is just a drop of blood to remind me that I'm talking to you about blood. This isn't actually stock art. If you look very, very carefully into this picture, you can see my stretched face and a couple of stretched faces of other people in the lab, whilst we were desperately trying to take a photograph of a drop of blood at the end of a pipette for something completely trivial, but so I won't worry you with that. Now, the way that clinical scientists and clinicians have been thinking about how to tackle the problem as it emerged last year, was to think about trying to understand how this particular virus causes the symptoms that it does, and how this might provide some clues and directions that we could use to develop new strategies to prevent them or to treat patients when they unfortunately are suffering from these particular symptoms. And the general approach, because of the timescales involved, has been preferably to use existing medication. And there's been some really big successes here. Probably the most significant one is dexamethasone, which is a well-used, well-practiced, been around for many, many years, a steroid drug that's used to suppress immune function. So it's commonly used as an anti-inflammatory drug. And as we'll see in a moment, COVID, the infection, causes severe inflammatory responses. So that makes perfect sense that this works so well. But various other things have been tried. We've got antiviral drugs, and in some individuals they're beneficial, but they're not sort of been as remarkable as may have been anticipated. We've got drugs that inhibit chemical messengers around that are present in the blood that orchestrate the immune response. And some other drugs have appeared that may interfere with the way that the virus enters cells, and hydroxychloroquine is a good example of that. And actually, it's another example of where really careful public health messaging has been important around this. So whilst we're investigating ideas and exploring how this might work, we really have to be very careful how we convey these messages to the public, because it could be jumped on for the wrong reasons. And well, we can remember Donald Trump's response to the hydroxychloroquine story. So before I tell you what we've been doing about blood clotting, I need to tell you where this fits into the whole story of COVID infection. And in order to introduce you to how this works, I need to tell you a little bit about how the virus infects your cells. Please bear in mind, I'm not a virologist, so be nice to me. So this is an example of a cell that has on its surface a receptor. It's called ACE2, angiotensin converted enzyme 2. And this protein lives on the outside, or it's a transmembrane protein, it lives on the outside of a range of different cells in our bodies. And like most viruses, they like to bind to a particular protein. They've evolved to bind well to a particular protein, which they effectively hijack and use as a way of attaching themselves to a cell. Quite often there are multiple steps, and in this case there's another enzyme. It's a protease which cleaves part of the protein on the surface that binds to the ACE2 and it also aids its entry into the cell. Importantly, the protein that binds to the ACE2 is the spike protein, which is the protein I highlighted in the first cartoon I showed you of this virus. This attaches the virus to the cell, which then fuses with the cell membrane, allowing the message from inside the virus to enter the cell cytoplasm. This then replicates and then causes more virus particles to be produced that then, of course, spread the infection. But as we'll come back to in a little while, the outside of the proteins that were on the surface of the virus often end up on the surface of the cell. And a cell that's producing virus packages those proteins that become the outside of the virus on the cell surface. So quite often we will see that virally infected proteins have got viral proteins on their cell surface. Sorry, virally infected cells have viral proteins on the cell surface. So we'll come back to this in a moment, but I just want to plant in your mind this ACE2 protein. So it's important to ask the question, where is this protein? Is it relevant to the places we know that the infection occurs? So these are some early post-mortem samples from patients following infection with COVID, and unfortunately they succumbed to the infection. And these are sections of the lung. They're different. This is a sort of blown up larger section here. On the right hand side, it's just this little part here which has been blown up larger. Now this is a type of immunohistochemistry where a stain has been used or an antibody has been used that labels ACE2, and it goes a dark brown color. So we can see here that there are where the arrows are, you can see evidence of ACE2 on the surface of what here is epithelial cells. So these are the cells that line the airways in your lungs. Is another example here. This is airway cells here, and you can see they stain quite nicely. These are epithelial cells. This is actually a cell line derived from epithelial cells from the lung, and again you can see they stain quite nicely. The other thing to point out up the top here is you can see that this little thing here is actually a little capillary in the lungs. So this is a blood vessel, and the arrow here, the arrowhead here, is pointing at an endothelial cell. So this is on the inside of the blood vessels. So just to remind you what blood vessels look like, going from arteries into capillaries and back into veins and returning to the heart. I'm not going to labour too much of this because it's not so important, the structures of these vessels, except to point out that they are lined with a single layer of endothelial cells. Remember, endothelial cells have this enzyme on the surface. So they're constantly lined. The inside of the blood vessels are entirely lined with endothelial cells, unless there's something wrong with that endothelial, with that blood vessel, such as injury. Underneath the endothelial cells, in red here, is collagen. So it's a basement membrane of collagen that's holding the endothelial cells to the blood vessel wall. And actually, collagens are part of the extracellular matrix that holds all of the tissues in the blood vessels together, including the smooth muscle cells that you can see here in the blood vessel wall. So the take-home message from this slide is that in the lungs, the cells that express or have on their surface ACE2 include epithelial cells, so in the airways of the lungs, and endothelial cells on the insides of the blood vessels. And of course, these endothelial cells go throughout the body. Wherever blood vessels go, this is where they are. So I've got a picture here that really emphasizes the complexity of the lung and why it's such a complex tissue, but also why it's so fragile. This is actually a photograph of an injection molding from the Cole Museum of Zoology at the university. And this is actually sheep lungs. And what you can see here is the lungs have been injected with 3 different colors of latex before the tissue was then digested away. The museum's got some amazing of these sorts of casts of all sorts of tissues, but this is probably the most beautiful. Basically, the airways have got a clear plastic in them. Blue is the veins and red is the arteries. And you can see the further we get away from the top of the lungs, the finer and finer the detail gets. And this is a blown up image of one of the tips of one of these areas just here. And you can see that the airways become tinier and tinier. They branch into these little bulbous ends called the alveoli, which are wrapped around by the blood vessels. So the blood vessels literally wrap themselves around these thin airways, which are very thin indeed. And that's how the gas exchange occurs between the blood vessels and the airways in the lung, of course, taking in oxygen and expelling carbon dioxide. So you can see that the lungs are really delicate and fragile, and inflammation or damage in these sorts of regions could compromise function quite easily. So if we home in and look inside one of these airways, what does it actually look like? What's going on in infection? So this is a cartoon of some alveoli. So these are the air sacs at the end of those little clear channels of plastic I was showing you just now. And in this case, there's a capillary wrapped around it. And so the very thin space here is where the gas exchange happens. It's aided by a surfactant that's on the inside of the alveoli. And you can see the flow of cells through this. So this is a functioning lung. In the case of a moderate infection with the virus, it causes localized inflammation. And this causes a number of different things. First of all, immune cells are recruited. So this is a macrophage, so it's there to eat bacteria or virus or infected cells, whatever. They get attracted to the site of infection. We find things become leaky and fluid starts to build up in the alveoli. And of course, that's going to stop the exchange of gas across this layer. We can see other things get released, such as inflammatory signals, which attract other cells of the immune system, such as neutrophils that are there to fight infection on the front line. And so basically everything becomes very glued up with this thick fluid, which effectively fills the space where the exchange of gases should occur. And effectively the lung fails. So you can see how really quite small changes can have quite dramatic effects on the functioning of the lung, and the lung's particularly fragile. So that's how we would describe this as an inflammatory response, but I'll show you in a moment or two there's some other things going on as well which involve some of the other components in the blood. There's another aspect to the infection which is really important, and you may have heard this referred to in the media, a cytokine storm. And this is really describing the immune system out of control. So immune cells talk to each other using chemical signals. things like interleukins in particular. And there's a range of other molecules here as well. And actually interferon gamma is a molecule that's involved in fighting viral infection. So that's also going to be relevant here in the response to the virus. But there's a whole range of these different chemical signals. And if the stimulus that's causing this to happen is too dramatic and too sustained, and this virus seems to be very good at doing this, then it leads to this sort of, it's like a chain reaction, and effectively we end up with a very, very dramatic and an overkill response, if you like, of the release of cytokines that should be coordinating the immune response, but really it just gets out of control. So it's referred to as a cytokine storm. And this is associated with severe inflammatory conditions. This is well characterized with a number of different viruses as well. Now, this is a picture from one of my earlier slides of the drugs that have been tried, and this is tocilizumab. Drugs have such stupid names, I do apologize. This is a monoclonal antibody that blocks the receptor for IL-6, and interleukin-6 is one of the key orchestrators of these really dramatic immune inflammatory responses in viral infections. So this has been tried and it is beneficial in some individuals, but again, not a wonder cure. So what's going on with the blood? Well, fairly early on, actually well before these stories started to appear in the media and in the scientific literature, clinicians started to notice strange things happening with regard to blood clotting and COVID patients. First of all, a large proportion of those patients who died from COVID and had postmortems performed had evidence of blood clots in their lungs, but also in other tissues, as we'll see in a moment. So clearly something strange was going on. Also, there are some of the signs that there are a number of different blood tests that are done to look at abnormal blood function, and things like evidence of blood clots can be measured using various assays that are used to measure soluble proteins in the blood. And there's some very strange signals in some of these fairly standard clinical tests that are done. And so people were scratching their heads trying to work out what's going on, because, you know, we have this viral infection that's causing the damage to the lungs, but for some reason, really quite substantial clotting is happening in individuals. And it wasn't just little clots in small vessels. Often it would be very large clots in very large vessels, which is very unusual. So where is this happening predominantly? Well, the key issue is clearly the lungs, because that's probably the most vulnerable organ in terms of compromising function and respiration. But clots are also found in the microvasculature in the kidneys. They're found in the intestines, in the portal vein from the liver. That's been reported quite a lot. And also in the heart. So in places where you might expect to have blood clots that are associated with heart attacks, frequently clots have been found, but with no evidence of atheroma, which is the normal thing that stimulates, or the abnormal thing that stimulates heart attacks due to the fatty deposits in blood vessels. Strokes have also been reported. So clearly a quite sort of distributed, strange clotting phenomenon. So many of the symptoms, as I've described, particularly the lungs, can be explained by inflammation. But what about blood clotting? You know, how is this contributing to this particular condition? I've mentioned most of this already. Many of the features look like a sort of broad disseminated coagulation around the circulation, elevated levels of deep vein thrombosis. Pulmonary embolism is quite prominent. That's where the deep vein thrombosis breaks away and lodges itself in small vessels in the lungs. And kidney and heart, I've mentioned already. So this started to raise the question as to whether we could target blood clotting as a way to try and prevent some of the more serious consequences of COVID infection, which seem to be associated with the more severe patients, or patients with more severe infection. And these are just a couple of, these happen to be from the Independent, but there are reports from a range of different places on studies that are underway looking at whether we could use, as they describe, blood thinning drugs or other ways to tackle this by specifically targeting the blood clotting process. So the answer to this question is we don't know yet. It's too early to know. And actually, whilst we have at certain points we've had an enormous number of patients, actually conducting clinical studies in this sort of environment is really challenging. It's been quite difficult. Maybe that's a conversation for another time. So we need to understand why why COVID-19 causes blood clots, because if we understood how this works, we're in a better position to try and prevent this. So I'm going to break a habit of a lifetime, and I'm going to tell you a half-baked story. I say half-baked, it's almost there. The first thing I'm going to talk to you about is actually in a manuscript that's been resubmitted in the last couple of weeks. So hopefully this will see the light of day very soon. And this is focused on the mechanisms of COVID-associated blood clotting and how this might be working. And also I'm going to tell you about an associated clinical trial that will be a consequence of, or is ongoing, but is a consequence of this work. To do this, I need to introduce you to some of my colleagues. As I'd rather do this as we go along in case I forget them. So Nikki Cooper is a good friend from many years, and she's a haematologist who works at Imperial College London. She also has a research lab there. And we've been talking and scheming over the last year and a half about various trials that could be designed to tackle different aspects of the disease and how this might be beneficial. Jeroen den Dunnen is from the University of Amsterdam, the medical school there, and you'll see the story, you'll see why we've teamed up with him over the last year to work on this particular study. Ian Jones may be known to some of you. He is a very eminent professor of virology at Reading, and as it happens, a coronavirus expert, which is kind of handy at this time. And Alex Bayer is one of my postdocs who's done some of the work I'm going to introduce to start with. Before I do this, I needed to give you a quick tutorial on how the blood clotting system works. So this works through two interrelated systems. One involves platelets, the tiny little blood cells in your blood that when they encounter tissue injury, stick together and cause the blood to clot. And also the coagulation process, which is a complex pathway of soluble factors that are in the blood that when they're triggered effectively turn the blood from a liquid to a gel-like substance. And this happens through polymerization of fibrin, which really turns this polymerization, turns the liquid blood into a gel, as I've mentioned. I'm not going to focus too much on coagulation. I'm going to focus more on platelets. But this is a sort of cartoon of how we know platelets function, principally in the arterial circulation, but potentially also in the venous circulation. So this shows a cross-section through a small blood vessel. And the features to remember are the endothelial cells, as I've mentioned already, that line the blood vessel. You can see a couple of red cells here. And these little purple things are tiny platelets. And you can see there's a little bit of rupture to this endothelial cell, and that exposes what lies underneath. Now, do you remember from the earlier cartoon, I showed you that underneath the endothelial cells is a layer of collagen. Now, platelets have receptors on the cell surface for collagen. In fact, the one that activates platelets when they see collagen was a receptor we discovered when I was a postdoc in Oxford. And this enables platelets to stick to the area of damage and then very rapidly become activated. And when they become activated, they become sticky. They bind to plasma proteins that glue them together. And you can see that literally they glue themselves together and plug the hole. And then the fibrin, the coagulation process starts to happen, and this just consolidates the clot and stops the blood from leaking. And this will eventually be cleared when the blood vessel has been repaired by macrophages. So the important thing to remember is that damage to endothelial cells, so when you cut yourself or when there's damage on the inside, or maybe when they become inflamed, exposes proteins that trigger platelets and coagulation. So damage to endothelial cells equals blood clotting, basically. The other thing to remember, or just to remind you, is that these cells are also where the ACE2 enzyme resides, okay? So that's the background to what triggers blood clotting. Now I'm going to come back to my colleague Jeroen in Amsterdam to tell you a little bit about why we started talking to him and his lab about the work they've been doing. So if you remember, this is the virus infecting a cell. So this could be an endothelial cell lining a blood vessel. The thing to remember is that infected cells have, like the endothelial cells, have spike protein on their surface, because either it's when the virus has gone in or when the virus is producing more virus particles, that's where the protein gets inserted, and then the new virus particles will bud from the surface of the infected cell. Now, of course, the spike protein is from an infectious agent, and our immune system is primed to generate antibodies against abnormal proteins. And so the immune system will create antibodies against the virus, but also against the proteins that are present on the surface of the infected cells. And certainly the antibodies that are produced in the immune system can potentially bind to these infected cells. Of course, this spike protein is actually the protein that forms the basis of the immunization system, of the immunization strategies that have been developed to combat COVID. And so this is an important part, this immune response is an important part of our response to the viral infection. So effectively what the antibodies do is they flag up foreign cells, or in this case, maybe infected cells, for destruction. And a really important cell in this respect is the macrophage. So this is a picture of a macrophage engulfing bacteria. And you can imagine that these bacteria may be coated in antibody. This is very much a stylized picture. It's not real, but you can, actually parts of it, probably electron micrographs, have been glued together and colorized. But the idea is that these are bacteria that are being engulfed by a macrophage, and these have probably got antibody bound to them, which flags them up to the macrophages to destroy them. So you can imagine if macrophages are able to engulf antibody coated abnormal looking cells, this could also include infected cells. So what's this got to do with COVID infection and antibodies? How does this all fit together? Well, Jeroen's lab discovered a really interesting feature of antibodies in the plasma of severely ill COVID-19 patients. So this is a cartoon of an antibody. It is an immunoglobulin G antibody. And I'm not going to focus on too much about how these work, except to say this end of the molecule is known the FC end of the antibody, and this is the FAB end of the molecule. And this is the end that binds to the pathogen, and this is the end, the FC end is the bit that binds to the immune cell to trigger an immune response. Now, the interesting thing about antibodies, and I have to admit, I really wasn't aware of this until about 18 months ago, antibodies are glycoproteins. Like many proteins have glycosylation. So they have these complex patterns of sugars that are added to them. And the thing that's interesting about COVID patients that are severely ill is they seem to have an elevated level of abnormally glycosylated antibodies against the virus, against the spike protein in their plasma. In fact, they have low levels of fucose and high levels of galactose. So it's low fucose, high galactose. Why is this important? Well, it turns out that this changes the way the antibodies work, and it makes them work better. It makes them activate macrophages better. So the macrophages become hyperactivated, and this potentially can lead to damage to the infected endothelial cells. This is the model, at least. And we would imagine that this would have an impact on triggering inflammation. And if inflammation causes damage to the endothelial cells, revealing the collagen underneath them, then this could also be a trigger for thrombosis. The other important thing here to flag up to you is how cells respond to antibodies. So cells that are equipped to respond to antibodies in your immune system have what's known as FC receptors on their surface. There's a whole range of different FC receptors, and that doesn't really matter. The principle is that there's a type of receptor that binds to the antibody, and the antibody is bound to the pathogen. So this antibody binding to the receptor triggers a response. It triggers cell signaling in the effector cell, in this case a macrophage, that triggers off its response, which in this case will be to engulf the pathogen or to damage or cause inflammation of the infected cell. The other breakthrough in Jeroen's story was that they identified which receptor was important for this. So we know which antibody receptor triggers this response in the macrophages. And what's more, we can target this because there's an enzyme that is stimulated by these receptors, the antibody receptors. And it's an enzyme that we know really well from our platelet work called spleen tyrosine kinase, or SIK for short. And this is a target for a drug that's been on the market for a very short amount of time, that's used to treat, well, various types of immune responses, and it's sometimes, yeah, it's various types of sort of hyperactive immune responses. And so this is really interesting because this is an existing licensed drug that inhibits an enzyme that we know is important for this elevated macrophage response, which might be causing a lot of the damage in the lungs. But what's this really got to do with the clotting? Well, it turns out that platelets also have an antibody receptor. We've known this for years, but we've not really ever known why they have it. But what's more, it's the right receptor. It's the receptor that's on macrophage, or the wrong receptor, depending on how you're looking at this. It's the same receptor that's present on the macrophages. And what's more, platelets also contain the target for the drug fostamatinib, SIC. So, and together, the receptor and this antibody are important, because when you stimulate, or when the antibodies bind to the Fc receptor on platelets, it triggers platelet function and clot formation. So we wondered, do abnormal antibodies that are present in the blood of COVID patients who are seriously ill, and these are the ones that have abnormal glycosylation, trigger platelet function? So the way we've approached this is using microfluidics. Well, actually, this is one approach with the data I'm going to show you. So what this is a tiny chip with a glass slide on the bottom of it. And there's a tiny little channel that runs through here. It's absolutely minuscule, but we know the dimensions of it. So when we flow, if we flow liquids through it, we can mimic the flow conditions that occur in blood vessels. So, and also what we can do is we can coat the inside of these channels with proteins. We would often use collagen because it looks like a damaged blood vessel. But in this case, what we've done is we've taken some spike protein that's produced in insect cells in Jones's lab. So he's been working on this for a number of different reasons. So this is the spike protein from the SARS-CoV-2 virus. We coat that on the slide, and then we expose the slide to antibodies. The antibodies might be normal antibodies from a person who isn't seriously infected, or they may be the abnormally glycosylated antibodies from someone who's seriously ill. And then what we do is flow blood over the top under the same sort of flow conditions that we might expect in big and small vessels. We've tried various different flow rates or shear rates, and we see what happens. Now, we need to be able to visualize this, and so we do this using a confocal microscope, which enables us to look in 3 dimensions and look at the depth of the blood thrombi that form. Now, obviously, blood's red. It's difficult to see the platelets because they're not red. So the way we do this is we stain we actually load the platelets with a fluorescent dye so we can see them. And because the confocal microscope uses fluorescence, that enables us to use this to visualize the platelet thrombi that form. So this is the first of a couple of data slides I'm going to show you. The top shows what happens under normal conditions and the appearance of the initial clots that form, or the platelet aggregates that form on this surface. So this has got spike protein on it. It's also got a protein called von Willebrand factor, or VWF. We've added this because this is one of the key proteins that traps platelets at sites of injury. And also, levels of this protein seem to be dramatically elevated in patients that have serious COVID infection. So this is a way of ensuring that the thrombi form. And each of these little green spots is hundreds of platelets. So the scale of this, platelets are tiny, so the scale of this is quite dramatic. Now, if we do the same assay with normal antibodies against the virus, not very much happens. It looks very similar to the control. But if we use the antibodies that have got the abnormal glycosylation, hopefully it's quite obvious here, there's a much stronger signal. We see much bigger, much deeper thrombi forming with far more platelets present. We can visualize this in the graph. So the bars to look at will be this one with WT stands for wild type, or the normal antibody. And this here is the abnormally glycosylated. So you can see it's quite a dramatic increase. Now you might ask, what's this 4.3 thing that's appearing on my graphs here? Because the 4.3 treatment seems to reduce it. It makes it go back to the sorts of levels it was in the absence of any antibodies. Well, 4.3 is a monoclonal antibody that's been designed to bind to the antibody receptor that's on platelets and macrophages, and to block it. So effectively, it blocks the function of this receptor. So when we block the function of the Fc receptor on platelets, the antibody receptor, we reduce, or we abolish the effect of the abnormal antibodies. So we can deduce from this that the antibody receptor on platelets is responsible for this particular process. We can look in a little bit more detail. So this is very similar data, and I'm just going to take you through pictures and the graph that goes with it. So this is a thrombi that form in the presence of the normal anti-spike antibodies from a non-seriously ill individual. And then if we look to the right here, this is using the abnormal low fucose, high galactose antibodies. So this is very similar to the graph I just showed you. You can see it goes up dramatically. Okay, if we just concentrate on the top now and look at these two here. So this again is the normal antibody, and this is what happens when we use this compound called R406. Now, it's a bit confusing this, but R406 is basically fostamatinib. Fostamatinib, so remember this is the drug that inhibits the enzyme that's responsible for how this receptor works. Fostamatinib is what's known as a prodrug. So it has to be metabolized by your liver. And this metabolism converts it into an active form. And so if we were to just use fostamatinib, as in the tablet that you would take, it wouldn't do anything. R406 is the active metabolite of fostamatinib. So if we add the R406 or the fostamatinib to the normal antibody treated, you can see nothing happens. And that's probably because the ordinary antibodies are just not doing anything. They're not enhancing the thrombus formation. But if we look at the similar sort of thing for the abnormal antibodies, you can see where we use fostamatinib, we reduce it back down, if anything, to below the levels it was at the start. So this indicates that using fostamatinib inhibits this abnormal thrombus, this abnormal blood clotting process that occurs, at least when we measure it in vitro. Now, there's some other abbreviations on here which stand for ibrutinib. So IBR is ibrutinib. This is an anti-cancer drug which targets another similar enzyme, actually. And this drug's used to treat various quite rare forms of blood cancers. And we've shown in the last couple of years that this also impacts upon platelet function and how that works and how important it is for platelet responses. And again, you can see it reduces the effect. And CANG stands for cangrelor, which is actually an antiplatelet drug that targets what's actually an ADP receptor on the platelet surface. And you can see this also reduces it. So this suggests that just by targeting platelets, we might be able, using existing drugs, we might be able to reduce the incidence of thrombosis associated with this viral infection and potentially others that are known to affect clotting. So the question arises, can we repurpose fostamatinib? So repurposing drugs is where you take an existing drug and you use it for some other purpose. Maybe because it does something completely different, or maybe because its target is involved in other processes. And I guess that's the case here. So can we repurpose this to prevent COVID-19 associated deaths? So this is work that's underway, and I don't have results for you yet, but I want to tell you what we're up to. This is relied really heavily on Nalina, Tanya and Joe, all members of my lab. Nalina's a postdoc, a very talented scientist. Tanya is a research technician who has worked with me tirelessly for 23 years, poor woman. And Joe Dunster is a mathematician. So we actually have a postdoc in the lab who's a mathematician, and that's been revolutionized the way we do a lot of our science because we have that maths and computational expertise. So this is a cartoon of a technique that we've developed, and I'm just going to very briefly mention it's designed to enable us to do clinical studies. And this is something that's been done really badly with platelet function in the past because it's such a difficult thing to measure, and it also because it's a cell-based assay. So we've developed a system that enables us to use a really robust approach. And what's more, we've automated all the software that analyzes the data for us, pulls out the appropriate parameters, and then we can use machine learning techniques to cluster the data and decide which group individual patients fall into. And this enables us to characterize an individual very, very precisely. This is very much an evolving science, but we're applying it to all of our clinical work at the moment because we think it's the way forward. So the way this is being used is in a clinical study that's been set up by my colleague Nikki at Imperial called the MATIS study, or the multi-arm trial of inflammatory signal inhibitors. Bit of a mouthful. Basically, what we're doing is recruiting patients who are hospitalized with mild or moderate pneumonia, COVID-associated pneumonia, and then they're randomized to 3 potential streams of treatment. One is the standard of care, and this is confusing because this keeps changing, but this would include things like dexamethasone and whatever has been, a number of other drugs that have been brought in over the last year or so. Fostamatinib, because we know this should inhibit our clotting, and it should also suppress the immune response. It should suppress the macrophage function. And there's some other related drugs that I won't mention today. And what we're hoping to ask with this particular study is, first of all, is clotting actually altered in COVID infection? Can we measure things in platelets that change? Does this get worse as the platelets decline? Does it get better as they recover? And actually an interesting feature of this abnormal glycosylation of antibodies is that this seems to revert back to normal as the patients recover, which is really unusual. And that's quite an interesting and unique property for this particular viral infection. We want to know whether fostamatinib can reduce the clots in the lungs that are, and elsewhere that are seen associated with these seriously ill patients. And also the role of these abnormal antibodies, because we now have access to lots of samples where we can extract the antibodies and see how they work, all from patients who've been infected. We want to know whether it reduces the inflammation. So my colleagues in London are looking at that. And of course, overall, we want to know whether it reduces the number of deaths. So this is underway. We've recruited 118 patients as of today. So it's going well, but it's going to be a little while before we can report on this because everything's blinded and we won't be able to unblind it until we've completed. So I hope maybe if I'm asked back in a couple of years, I'll be able to tell you what happened and report where this led us. But I'm hoping it will change clinical practice and will also tell us a lot about how viruses impact upon the immune system, but also how that triggers abnormal clotting, which is a problem with a number of other conditions. So if I'd been giving this talk like 3 months ago, that's probably where I would have stopped. But if I've got the time, I've just got 1 or 2 more slides. Is that all right, Ken? I just wanted to, yeah, good. Because there's another study that's tended to occupy a lot of my, or a lot of, an effect that has started to occupy a lot of my time in the last few months. It was probably about beginning of April, some rumours started to emerge through the media that I got sort of early warning of, and about potential issues with one of the vaccines causing clotting. And for the weeks that followed this led to being overwhelmed by media and journalists and TV and that sort of thing, which fortunately has now calmed down a little bit. But it was a really interesting time, again, to be working on a subject that everyone's interested in, because it was suspected that the effects that were being seen were due to effects on platelets. So this was a report that appeared 3rd of April, where the first clots, in the first blood clot deaths after the AstraZeneca vaccine were reported in Europe. And this included the UK. And to be honest, it was tough to really work out whether this was something real or not at the outset. The thing to really point out is it's very rare, and that made it very difficult to study, and still makes it very, very difficult to study. But the thing that really made it stand out was that it was a particular type of blood clot that seemed to be quite noticeable. It still was very rare, but it was occurring in people you wouldn't expect it to happen in, particularly the young. Seemed to also be a sort of bias towards women as well. But this is a type of blood clot in the brain called the cerebral venous sinus thrombosis, or CVST. And this sort of set off alarm bells because this isn't something that's seen very often. But that in itself is a problem, because if something's very rare, then probably it's underreported because it's not diagnosed properly as often as it maybe should be. It was also complicated by the literature that had all sorts of estimates for the actual incidence in the normal population, which ranged from 1 or 2 per million per year to 17 or 18 per million per year. And that makes a big difference when you're looking at such tiny numbers to try and work out what's really going on. The other thing to say is that you would, in normal times, we wouldn't really be able to make this association with a new drug or a new treatment so quickly. The fact is that because so many people are being immunized so quickly over such a short period of time, then these signals start to emerge that maybe exist with other vaccines, but we just don't know because of the way the studies are done over much longer periods of time, or far fewer people actually having them. But clearly something was going on. And actually this has a name now. It's called vaccine-induced immune thrombocytopenia. Thrombocytopenia refers to low platelet counts, and this can be dangerous because it usually causes bleeding. Clearly this is a bit unusual because this is a thrombotic disease and it's immune driven. So it's vaccine-induced immune thrombocytopenia. Now we know it's immune driven because of the time, we suspected initially at least, because of the timing when this starts to appear. It happens about the time when people start to develop immunity to the vaccine. And actually, it's an interesting parallel that the clotting that people have with COVID infection appears at about that same time, which again fits with the idea that that's an immune antibody-driven effect. The other thing that I should perhaps also flag up is the fact that this signal was seen with the AstraZeneca vaccine, which uses adenoviral vector, which I'll mention very briefly in a moment. And this specific pattern, this type of clotting, wasn't seen or hasn't really been seen with Pfizer or Moderna vaccines. Obviously, Pfizer's been used more than Moderna. Other clots have been seen occasionally, but probably but not this type and not this sort of level. So what causes VITT, as it's been called? Well, this is Mary Scully from University College Hospital in London, and she had a bit of insight and actually really made a bit of a big breakthrough with this. It wasn't her breakthrough really wasn't to think something that other people hadn't been thinking. She just went a stage further and actually tested it. She looked at the symptoms of these individuals, and she'd only seen one or two, and then there were obviously other reports in the literature, and thought, these look like a different disease. These look like another condition, but there are aspects of this that just don't make sense. But nevertheless, I'm going to do the tests anyway. So she sent samples off to the lab, and sure enough, there were some features of this other condition, but not all features. The other nice thing about this is that she was recently awarded an MBE for presumably for this, but also her other prominent work in the hematology area. So the condition that she was thinking about is a condition known as heparin-induced thrombocytopenia, or HIT as it's known. And this is really not a very well properly characterized disease, but we know quite a lot about what triggers it. It's life-threatening, and it's caused by low platelet counts, but just like the clots that form in the vaccinated individuals, is also thrombotic. So this sounds like a sort of, it sounds like I'm making no sense here, because this is a condition where individuals clot and they bleed, and this makes them very difficult to treat. These particular HIT patients are really difficult to treat because they're bleeders, but they've clotted first. And the reason they bleed is because their platelets have basically been consumed in clots. So the way this works is, you imagine some platelets become activated. When they activate, they release all sorts of chemical messengers. I haven't really talked about that, but it's one of the major things they do. And this sends signals to other cells. One of the things they release, which is actually anticoagulant, is platelet factor 4, or PF4, as it's in these little green things here. You can see it's spitting some out. Now, PF4 sticks back onto the surface of platelets, and it sticks to membranes of other cells. But it also sticks to a molecule known as heparin. And heparin is ironically, an anticoagulant drug that's very successful, used very commonly. But for some reason, in some individuals, heparin binds to platelet factor 4. So they bind together. And when they bind together, this changes the shape of the platelet factor 4, this protein. And the body sees this as being a foreign protein and generates antibodies to it. So it generates antibodies to its own protein. So these are autoimmune antibodies. And actually, this whole thing is usually on the surface of activated platelets. So what this does is this antibody then binds to the antibody receptors on platelets and activates them. And so you get this massive activation of platelets in the circulation, and it's triggered by an autoimmune response that in itself is triggered by heparin, this drug, that somehow changes the conformation of this normal protein that makes it immunogenic, as we'd refer to it. And the result of this is that you have thrombosis, you have horrible, nasty blood clots form in the circulation. And this consumes the platelets, which means the platelet counts of those individuals are very low, which then puts them at risk of bleeding, which makes them very difficult to treat, as I've said. Now, the really interesting thing is that the COVID infection looked like this. It's very similar, except most patients who have the vaccine-induced thrombocytopenia have never seen heparin. And that's the difference. And that's what we don't quite understand. However, it seems that most vitt patients have anti-platelet factor 4 autoantibodies in their plasma. So for some reason, which we don't understand at all, they've generated autoantibodies against this protein that causes symptoms that look like heparin-induced thrombocytopenia. We don't know why why some people it happens to and others it doesn't. Clearly, it's very, very rare. So that might mean it's a gene polymorphism in some individuals, makes them more likely to generate antibodies. They may have underlying conditions. It may be molecular mimicry. There may be something about the virus proteins that look a bit like platelet factor 4 in some individuals, and therefore antibodies against the virus look like proteins, are directed against proteins that they shouldn't really bind to. So this is how this sort of general theme is developing at the moment, and it's what we understand of how this clotting works. This is a recent preprint from a colleague Pip Nicholson in Birmingham, and basically what they show in this paper that they say I should point out this is on 4 patients. These are all on very small samples because the numbers of patients are very low, and getting hold of patients' samples is really hard. But it says all patients tested positive for anti-platelet 4 antibodies despite no prior heparin exposure. Serum from patients with vitt but not healthy donor controls induced platelet aggregation, which was abrogated following IVIG. That's just injecting antibodies into individuals. It's a way of fooling the immune system that's commonly used as an anti-inflammatory response treatment. But also aggregation to platelet sera was blocked by 4.3 Fab. That's the antibody we were using earlier on to block out antibody receptor, which targets Fc gamma R2A, the antibody receptor that we target. And inhibitors of, and amongst this list is SYK, the kinase that we've been working on, and also BTK, which was the target of the other drug that was in one of my slides earlier on that we've been working on. So it's very interesting. There's some really strong parallels here between what's happening in COVID-induced thrombosis and what's happening in vaccine-induced thrombosis. There are some clear parallels there. And those really lie around the antibody receptor. This antibody receptor seems to be really important in generating the signals that cause the response. But the good part of this is that we do have ways of inhibiting this, whether that's broad antiplatelet therapies like simple aspirin, or whether it's some of these other more exotic drugs that have been more recently developed for cancer and immune response problems. The other thing we also know with patients who have any sign that they may have abnormal clotting following vaccination is that they must not be given heparin, because that may exacerbate the problem. Again, we don't properly understand why these individuals have this response when they haven't seen heparin, but it's inevitable. It's going to make it much worse if they're given this anticoagulant. And it's a common anticoagulant to give people if they've got these sorts of abnormal clotting. So I guess just to finish, I wanted to just mention where we are with the vaccine side of things because of these risk factors. And there's a couple of infographics here. The one on the left is from the BBC, and it really shows your chances in a million of serious harm due to vaccine side effects. In a 25-year-old, it's 11 in a million, whereas in a 55-year-old, it's 4 in a million. So the risk seems to be higher in the younger population. But your risk of dying with coronavirus infection is much greater in the older than it is in the younger, which means it tips the balance of ratio of risk benefit balance against giving these sorts of vaccinations against the young, because the benefit is much lower. This is an infographic from one of Chris Whitty's one of his press conferences, which I think showed really nicely. It's in the same sort of way. So on the left is potential benefits, and on the right is potential harms. This is the effect of the AstraZeneca vaccine. And you can see in the older generations, the benefit is very high, the risk is really low. And whilst the numbers are really, really small, the risks of serious harms of the vaccine are higher in the young, but the risks, the actual potential benefits of the vaccine, because they're much less likely to become seriously ill, are much lower. And so because of this, the recommendation now is that under 40s are offered the Pfizer-BioNTech or Moderna vaccines. But if they've had the AstraZeneca as the first round, then they would have the same one again after this. I didn't want to end on a sort of negative sounding point, so I just wanted to really flag up the fact that the vaccination program has been an overwhelming success. This is, as of today, the cases in the UK the whole of the UK, and you can see numbers are starting to creep up again, and this is worrying, and that's inevitable as we take the brakes off. But if you look at the death rates underneath, it's maybe slightly creeping up a tiny bit, but it's way behind where we would have expected a few months ago. So clearly there's been some benefit there, and hopefully this will go from strength to strength as more of the population become immunized. And I thought this was really nice. This was at Wimbledon a couple of days ago. Sarah Gilbert got a standing ovation for her role in producing the vaccine, amongst a team of lots of other people. So my last slide is just final thoughts, just to remind you what I've been thinking about here, talking to you about. The fact that COVID-associated thrombosis is really serious. It underlies many of the really serious side effects, and probably contributes to pathology, certainly pathology, and certainly probably mortality. But we also have this emerging story of vaccine-associated clotting effects. But there's a common theme with these, and these are the antibody receptors, whether they're on macrophages or whether they are on platelets. And the important thing is we have potentially the ability to target this mechanism and this receptor, and this may be beneficial in preventing some of these really quite unpleasant and life-threatening side effects or effects of infection. And with that, I should just acknowledge my colleagues who I've mentioned as we've gone through. I must mention the British Heart Foundation. They allowed us, in fact, they encouraged us to misappropriate some of our funding last year so that we could direct it into this work. And then more recently, we've got funding from the Medical Research Council to continue it. So hopefully we will get some interesting answers for this that will be beneficial, not only for this story, but maybe other viral infections.